So in this video, we're going to cover the plaque assay, which is activity 6-5. And we're going to start by talking about day 1, which is the setup for the plaque assay. So before we can talk about our plaque assay, we need to discuss about how we set up this experiment. So the purpose of the plaque assay is to determine the titer of a virus, meaning that we want to know what is the concentration of this unknown stock of virus. So to do that, we need to do something similar to what we did in our standard plate count. And in our standard plate count, remember that the purpose of our standard plate count was to determine the titer of a bacterial stock. So to determine the titer of bacteria. In this case, we're determining the titer of a virus. And in this experiment, the type of virus that we're using is a type of virus called a phage. And a phage is a virus that infects bacteria. So to start this experiment, the first thing that we have to do is we need to do a serial dilution. We need to make our serial dilution of our phage stock solution. So to do that, we are going to start with our stock solution. Here's our stock solution. And we're going to transfer 0.1 milliliters into a volume of 9.9 milliliters of this tryptic soy broth. So this is just a rich media. So in this tube, we have 9.9 milliliters of TSB. And so when we add our 0.1 into 9.9, that brings our total volume of 10. So we have 0.1 milliliters into a total volume of 10 milliliters. So what that works out to is, if we do that calculation, that is the same as 1 over 100. Basically, we're moving our decimal point one place to the right. So if I move it one place to the right on the top, I need to do the same thing on the bottom. And that's where we get this other 0. So now we have 1 over 100. That is the same as 1 over 10 to the second. This is a 1 with 2 zeros. So 1 over 10 to the second. And if we do that math further, what we're going to get is 10 to the negative 2. So remember that if we take 1 over 10 to the second, when we divide 1 into 10 to the second, it now makes this a negative exponent. So this is now our negative, or 10 to the negative second. So my first tube has a dilution factor, or in this case, our final dilution, of 10 to the negative 2. We have diluted it 10 to the negative 2. Now, we're going to take 0.1 milliliters of this, and we're going to transfer again into 9.9 milliliters of the tryptic soy broth. So we're starting with 10 to the negative 2. Our dilution factor, remember this is our dilution factor. This is the amount we're diluting each time. Our dilution factor here is still 10 to the negative 2. So we just add our exponents. 10 to the negative 2, 10 to the negative 2. My final dilution in this tube, 10 to the negative 4. Then we take our 10 to the negative 4 and we transfer 0.1 milliliters into 9.9 milliliters of the tryptic soy broth. So again, we're starting with our 10 to the negative 4. We're diluting 10 to the negative 2 more because this is my dilution factor. Add my exponents. My final dilution in this tube, 10 to the negative 6. If you're struggling with that math, go back and review the video for the standard plate count. I went through this in a lot more detail. Now, remember, this is our virus stock solution, right? These are my serial dilutions for the virus. Now, if you think about a virus, remember that viruses are obligate intracellular parasites. They can only replicate within a host cell. So I can't just take that virus and put it on a plate. If I do that, the virus is not going to grow. It needs a host cell. So in this case, our host cell is going to be a strain of E. coli called E. coli strain B. So when we go to pour our plates, not only do we need to add the virus, but we also need to add E. coli to each of our tubes before we pour it. So if we start with our 10 to the negative 4 and we transfer 1 milliliter, remember that my final dilution factor is still 10 to the negative 4. Because my dilution is per milliliter. So if I transfer 1 milliliter, my final dilution on that plate is still going to be 10 to the negative 4. If I take my 10 to the negative 4 and I only plate 0.1 milliliters. 0.1 is tenfold less. It's moving the decimal point one place. So what that means is instead of 10 to the 4, negative 4, now we have 10 to the negative 5. Then we have this one here, 10 to the negative 6. If we plate 1 milliliter, our final dilution on that plate is still 10 to the negative 6. And then lastly, if we look at this bottom plate, you can see that my final dilution on this plate is 10 to the negative 7 because I'm taking my 10 to the negative 6 and I'm plating 0.1 milliliters. And when I plate 0.1, it's tenfold less, which is my 10 to the negative 7. Now, in addition to adding the virus, again, I'm going to add E. coli. I'm going to add the host cell. And to each of these tubes, I add 0.5 milliliters of the E. coli. Now, notice that that amount of E. coli does not affect my final dilution factor. That's because it's not diluting the virus anymore. I am pouring this entire content onto this plate. So my final dilution is not influenced by the amount of E. coli. I'm just adding a constant amount of E. coli to each of these tubes so that I get what we call a lawn. I end up with so much bacteria that basically the whole plate is covered. And you're going to see why in a little bit to see why that's so important. But so this would be the first part of your experiment, would be to do your serial dilution. And again, this is how we do our calculations. So here is the overview of what we are doing in this experiment. So again, you would start with your serial dilutions, that's this top part, using a micropipetter. And remember that if we're transferring 0.1 milliliters, that's the same as 100 microliters. So we are going to start by setting up our serial dilutions. Now, once we set up our serial dilutions, then we need to make our tryptic soy agar tubes. And again, we're only going to take one out of the water bath at a time. And the reason for that is that remember that these tryptic soy agars, they are kept in a 55-degree water bath. And they're kept at 55 because once that agar gets to 42 degrees, it's going to solidify. So we want to keep it at a higher temperature to keep it liquid. So we take out one tryptic soy agar at a time. And so for example, if I take out this one, I need to add 1 milliliter of this virus solution, and I need to add 0.5 milliliters of the E. coli B. So I need to add both to this tube. I will mix, and then I will pour over these tryptic soy agar plates. And when you do this, you'll see that these plates already have a thin layer of agar on the bottom. And we're going to pour this agar over the top. We have to work quickly so that the agar doesn't solidify. And so when you see me do this in the video, you'll see that I do this fairly quickly so that the agar doesn't solidify. Once I pour my plates, I don't want to move my plates because if I move it, it causes the agar to slosh around. And it's going to contaminate the lid, possibly the desk, and also your virus concentration is not going to be consistent. So you have to make sure that once you pour these that you don't move them. Also, when pouring these plates, you want to make sure that you pour them near the flame because your flame is your sterile field. And so you'll see that in the video that when I do this, I pour it near the flame. You will do this for all 4 plates. Once you have your 4 plates made, you are going to rubber band those plates and then you're going to place it in the incubator. And so this assay would be done in pairs. You would do this with a partner. And so each pair would need the following materials. And again, this is shown in the video. So each pair would need 3 tryptic soy broths. Those are to do your serial dilutions. Each pair would need 4 TSA overlay plates. So again, those are the thin TSA plates that already have some agar, and then you're going to add more to it. Each pair needs 4 TSA soft agar melted deeps. And again, we're going to keep them in the water bath at about 55 until we're ready to pour that tube. We need a sterile 1 milliliter transfer pipette. That's used for pipetting the bacteria. We can use those transfer pipettes, those plastic ones, because we don't need accuracy so much. The bacteria is just there to be a lawn. If the amount is not perfect, it's not really going to matter. We do, however, need to accurately pipette our phage, which is our virus that we're trying to determine the titer. So because we need to be accurate with our pipetting for the phage, we need a P1000. So you're going to have a P1000, 1 per pair. You need 1 box of sterile P1000 micropipette tips, the blue ones. You would need a 600 milliliter plastic beaker for used tips, so once your tips are contaminated, to dispose of them. You would need 1 virus suspension, which is your phage that you're determining the titer, so you would have 1 per group. And then you would have your E. coli strain B, 1 per group, because again, when we want to determine the titer of the phage, we have to also add the host cell in order for us to see where the virus is. The virus will not replicate without the bacteria. And so the next part of this video is going to be demonstrating the technique so that you can see how this experiment is actually done. So in this experiment, I'm going to be demonstrating how to do a plaque assay. And so the purpose of a plaque assay is to determine the titer of a virus. And in this experiment, we are determining the titer of a type of virus called a phage. Phage is a virus that infects bacteria. So what we have is we have our unknown phage solution, and this is what we want to determine the titer of. You may recall that we did something similarly when we did our standard plate count, and in our standard plate count, we were trying to determine the titer of a bacterial solution. This time, we're trying to determine the titer of the phage. Now, remember, the viruses are obligate intracellular parasites. They cannot reproduce outside of a host cell. So we can't simply just put this on a plate to determine the titer. It's not going to grow on its own. So in addition to having phage, we also have a strain of bacteria. We're using this E. coli strain B. And E. coli strain B is a strain of E. coli that is more susceptible to phage infection. And so this needs to be mixed with our phage in order for our phage to replicate and for us to determine the titer. So we need our phage solution, we need our E. coli solution, we need our micropipetters again, we need our micropipette tips, we need a used tip waste container, we need 3 tryptic soy broths, and these tryptic soy broths are what we are going to use to do our serial dilution. Just like we did a serial dilution for the standard plate count, we are going to repeat that process, but with the phage. We're going to do a serial dilution with the phage. So we have these 3 tryptic soy broths. This is what's going to be used for our dilutions. I have a transfer pipette, and so this transfer pipette is going to be used when I need to transfer E. coli because I don't need that amount to be very accurate. So that's going to be for this. I have my Bunsen burner, I have my lighter, and so I also have my 4 tryptic soy agar plates. And so for these tryptic soy agar plates, these are going to be thin plates, and we overlay the agar over this. So we have 4 of these. So I'm going to start out by turning on my Bunsen burner and getting that going. And I'm also going to label my plates. So for my plates, I need to label this with my initials. So JW. I'm going to put the day and time. So let's say I'm in the Monday, Wednesday, 3 p.m. lab. And then I'm going to put the titer on here. So I have my 10 to the negative 4. That's 1. And I'm going to put the date. So today's date is going to be 4/30/21. And I'm going to repeat that for all 4 plates. Except this is my 10 to the negative 4. This is my 10 to the negative 5. I'm going to label this one 10 to the negative 6 and 10 to the negative 7. And then I would label those completely with my name, lab day and section, and the date on all 4 plates. But for the sake of time, I'm not going to do that. I'm also going to label my test tubes that have the tryptic soy broth because this is what I'm going to do my serial dilution. So one of them is going to be labeled 10 to the negative 2. I have my 10 to the negative 4. And I have my 10 to the negative 6. So I've labeled all of the supplies that I need, and I'm going to start by turning on my Bunsen burner. So I need to turn on my gas. Remember, the way that I do that is I like to start with my baffle being open, and that allows the air to go through. So I start with the baffle open. I turn on the gas until I hear a hissing sound. Once I hear that, I close the gas a little bit. Still too high. So we have a little bit of a draft in here. There we go. So there's our Bunsen burner lit. And so you can see that we have our inner cone. That is the hottest part of our flame. So we need that in order for us to sterilize items. First thing I'm going to do is I'm going to change the volume on my micropipetter so that I'm transferring 100 microliters into these tryptic soy broths. So I'm going to open and release the micropipetter so that I can change the dial. And I'm going to change this, and I'm trying to set this at 100 microliters. And so on my micropipetter, it's going to read 0100. So after I set it and it's reading 0100, I need to lock it. Remember that when I do this experiment, I want to keep my tip closed, these tips closed as much as possible. I only want to open them when I go in to grab a tip. So I keep this closed as long as possible to keep these sterile. Now, remember that when we use our micropipetters, here is the plunger, and our plunger has a first stop and a second stop. Remember that the only time that we go to the second stop is our blowout phase, when we're trying to get all of the liquid out of our micropipetter. So what I'm going to do is I'm going to open my tips, and I'm going to get a tip on my micropipetter. So now I have my pipette tip. I am going to take my phage, this is the one that I'm going to do my serial dilution, and I'm going to vortex to mix. I can either do this manually, or I can do this using an electronic vortex. Either would work. And so I need to start by transferring 100 microliters of my phage stock into my 10 to the negative 2. This dilution in this tube is going to be 10 to the negative 2. So I vortex this. I'm going to take the cap between my pinky and ring finger, and anytime I go into a tube, I need to flame the opening. Push to the first stop, put my tip in, release the plunger, come out, flame it, cap it. Now notice how I go under my pipette and not over it. I don't want to go over my pipette because that could contaminate my sample. Now I need to add it to my first tryptic soy broth. So I'm going to take the lid off, flame it, add it, push all the way to the second stop to get all of that liquid out, flame it, cap it. I'm going to move it in my rack so that I know that it's done. My tip, I'm going to eject in my tip waste. So I'm going to get rid of my tip. Before I transfer from this tube to my 10 to the negative 4, I need to vortex. So I'm going to vortex to mix well. So that way my virus is completely suspended. Okay, so I've mixed that well. I'm going to get a new tip. So here is my pipette with the new tip. I'm going to take my tube, cap between my pinky and ring finger, take the cap off, flame it, push to the first stop, release my thumb to get the liquid in the tip, flame it, go under. I'm done with that one. I'm going to add it to the 10 to the negative 4. Cap between pinky and ring finger, flame the opening, push to the second stop. We want to get all that liquid out of the tip. Flame it, cap back on, put that here, eject, get a new tip. So I get my next tip. Now I'm going to transfer from my 10 to the negative 4 into my 10 to the negative 6. So again, I'm going to vortex before I take any liquid out. I'm just going to give it a little hand vortex as well. Okay. And again, I'm going to go under my pipette. So cap between my pinky and ring finger, flame the opening, draw up 100 microliters, flame it, recap. I'm done with this one. I'm adding it to my 10 to the negative 6. Take the cap off, flame the opening, add it, push to the second, stop, flame it, cap, and get rid of my tip. So now I have my 3 serial dilutions. I have my 10 to the negative 2, 10 to the negative 4, 10 to the negative 6, and so now I'm ready to use these on my plate. So like my standard plate count, when I do this experiment, my 10 to the negative 2 is not going to be plated because my lowest dilution on my plate is 10 to the negative 4. So for your 10 to the negative 2, you can just move that off to the side. You don't need it. The only reason you needed it was to make your 10 to the negative 4 and then make your 10 to the negative 6. So my 10 to the negative 2, I'm just going to set aside. I don't need that. But I am going to need my 10 to the negative 4 and my 10 to the negative 6. So notice that my first plate is labeled 10 to the negative 4. So remember that if this dilution is 10 to the negative 4 and I plate 1 milliliter, if I plate 1 milliliter, my dilution is still going to be 10 to the negative 4. So I have to plate 1 milliliter of my 10 to the negative 4 onto my 10 to the negative 4. For my 10 to the negative 5, I need 10-fold less. So for that one, I'm only going to transfer 0.1 milliliters, 10-fold less. Remember that my 0.1 milliliters is going to correspond to my 100 microliters. Since I already have my pipette set to 100 microliters, I'm going to do my 10 to the negative 5 and my 10 to the negative 7 plate first, because those are the ones that are going to require the micropipetter to be set at 100. Now, when I do this, these are thin plates. They're thin auger plates. So what I have to do, just like in my standard plate count, I have a melted auger deep. And my melted auger deep are kept in my water bath, and they're kept at 55 degrees. And the reason for that is that the 55 is going to keep it a liquid. When the temperature gets down to 42, that auger is going to solidify. And so I need to keep these in the water bath until I'm ready to do this. Now, let me explain what I'm going to do for my 10 to the negative 5. For my 10 to the negative 5, I have my serial dilutions. And again, I'm going to take 100 microliters from my 10 to the negative 4. And I'm going to add it to the melted auger deep. But again, I can't just add my virus, because if I add just my virus, then it's not going to grow. So in my melted auger deep, I'm going to add the virus, and I also need to add my E. coli. So this is my E. coli. Again, I'm going to vortex this, just like I did for the phage. So here it is. To transfer the E. coli, I'm going to use a plastic transfer pipette, because the pipetting of this E. coli does not have to be exact. The purpose of the E. coli is that when we mix the E. coli with the melted auger and we pour it, the E. coli is going to form a lawn on that plate. And then the virus is going to infect that E. coli. So for the purpose of this, for my E. coli, I can simply use my transfer pipette. And my transfer pipettes, these are 1 milliliter transfer pipettes, and I need to transfer half a milliliter of the E. coli to the melted auger deep. So I'm going to transfer both my phage and my E. coli to a melted auger deep, and then I'm going to pour it. Now, remember that when I pour plates, my sterile field is around my flame. Because remember that this area, if things are to fall and it hits the flame, well then it's going to help keep it sterile. So I'm going to move those close to my flame. In addition, the first one that I'm going to do is going to be my 10 to the negative 5. I'm ready to turn this one so that the auger side is down, so that I'm pouring the liquid on top of the auger that's already in there. I don't want to pour the liquid into the lid. So I'm ready now, and I'm going to take out one of my melted auger deeps. And I'm just going to wipe off some of the excess water from the water bath, just so it doesn't drip all over. So this is my melted agar deep. So this is my agar deep, and I'm going to put this here. I need to add both the virus as well as the E. coli. So I'm going to take my pipette, and again, I'm going to vortex this one before I go into it. I'm going to go under, I'm going to flame it, I'm going to draw up 100 microliters, flame it, close it, put it aside. I'm going to add that to my melted agar deep. Flame the opening, add it. Push all the way to the second stop to get all the liquid out. Take your tip, eject. Now I need to add my E. coli. Again, I'm going to add my E. coli using my transfer pipette. So I'm going to open this. This is sterile. So I'm going to open this. If I can get this to separate. All right, so I'm opening this up. And again, I want to keep this as sterile as possible, so I'm keeping it in the wrapper until I'm ready to use it. On my transfer pipette, there's a 1 milliliter mark, and then partway down there's one that's 0.5. That's how much of the bacteria that I need to transfer. So I'm going to take out my transfer pipette. Before I do that, I'm going to re-vortex my E. coli because I want to mix that. Take my cap between the pinky and ring finger, and I'm going to flame the opening of the tube. I'm going to take my transfer pipette, and I'm going to put it in my E. coli, and I'm going to draw liquid up to the 0.5. Now I let that go. I'm going to flame it, cap it, and I'm going to set it here for a second. Now I'm going to add it to my melted agar deep. So flame it, add. Dispense all the way. Flame it, cap it. And I have to work quickly at this point so this doesn't solidify before I pour it. I'm going to put my transfer pipette back in the wrapper until I'm ready to use it again. So I have my melted agar deep. It has my virus, it has my bacteria. Now it's ready to be poured. So when I do this, I like to flame the opening of the tube, and I like to let it cool a little bit because I don't want this bacteria and virus that's in my melted agar deep to touch that hot rim of that tube. So I just kind of let it sit for a minute, and then I'm going to pour carefully over the agar and try and get all of the agar out of that tube. Okay, and then I'm going to recap this. And now I'm done with this tube. So I can put that aside. Once I pour that plate, I want to make sure not to move it. Because if I move it, the agar is going to slosh, and you're not going to end up with a nice even distribution of the E. coli with the phage. So once that's poured, I'm going to leave it. Again, I have my micropipetter set to 100 microliters. So I'm going to do my 10 to the negative 7. But this time, to do that, I'm going to draw from my 10 to the negative 6. So before I start, I'm going to vortex my 10 to the negative 6. I'm going to re-vortex my E. coli because I'm going to use that again. I'm going to get another melted agar deep. I'm going to wipe off the extra water from the water bath. So there I have it. Again, I have to work quickly before that solidifies. So I'm going to get a tip. And again, I'm taking from my virus dilution. And I'll just go ahead and re-vortex again. Take the cap between my pinky and ring finger, flame it, go in, draw up my 100 microliters. Flame it, cap it, and I'm going to add that to my melted agar deep. Flame the opening, add it in, push to the second stop to get it all out, flame it, cap it, get rid of my tip. I need to transfer my E. coli, so I'm going to use my transfer pipette again, and I'm going to pull it out. And again, I'm going to vortex to mix. I'm going to flame the opening, and I need 0.5. Okay, and then once I have that measured, I can let it go up in the pipette. It's not going to hurt it. Flame it, cap, add it to my melted agar deep. Flame it. And I want to make sure not to touch the transfer pipette to the tube that has the virus. So flame it, put the cap back on. Then I'm going to put this back in here. If I touch this to the tube that has the virus, now this E. coli stock is going to be contaminated. So take care when you're transferring with your transfer pipette. Make sure not to touch the contents of the tube when you go to add it to this tube. Now I need to pour it, so I'm going to turn over my 10 to the negative 7, and I'm going to vortex to mix very carefully. I don't want to create too many bubbles. I'm going to flame the opening, and again, I'm going to let it cool for just a minute so that the opening of the tube is not too hot to kill the virus or to inactivate the virus and to kill the bacteria. And then I'm going to pour this. And then I'm going to recap this, and I'm done with this one. So now I've plated my 10 to the negative 5 and my 10 to the negative 7. Now I need to change my pipette to read 1,000 microliters. 1,000 microliters is equal to 1 milliliter. So I'm going to open this, and I'm going to set it to 1,000. And then now I'm going to lock it. Now I'm ready to get another agar deep. And again, I'm going to dry it off so that I don't transfer water all over from the water bath. I'm going to put it here. I'm going to put a tip on my pipette. And I'm going to go back to my 10 to the negative 4. Because my 10 to the negative 4, I need a plate on my 10 to the negative 4. If I transfer 1,000 microliters, that's equal to 1 milliliter, which means my dilution stays the same. So I'm going to vortex to mix this before I go in there. Take the cap between my pinky and ring finger, flame the opening, put my tip in, slowly release my thumb to draw the liquid into the tip, flame it, recap it. I'm going to add that to my agar deep. Flame it, pipette it in, flame it, cap. Now I'm ready to add my E. coli, so I'm going to vortex again. And remember, I'm using my transfer pipette, so I'm going to get back out my transfer pipette. And I'm going to flame it. And I'm going to draw up 0.5 milliliter. Okay. Flame it, cap, add it to my agar deep. Flame. Again, don't touch the transfer pipette to the agar that has the virus in it. Flame it, cap, set it down. Put this back in the wrapper. Now I'm ready to pour it on my 10 to the negative 4. So I turn it over. I'm going to vortex this to mix it gently. And then I'm going to flame this. And I'm going to let it cool before pouring it. But when I'm letting it cool, I want to keep it close to the flame because that's my sterile area. So once I let it cool, I'm going to open my plate. And I'm going to pour the agar on top of the agar that's already in there. Trying to get it all out. And then cap this, put it aside. I have one plate left, and that is my 10 to the negative 6. So I'm going to repeat this again. So I'm going to take out my last melted agar deep, and I'm going to dry off the extra water. Just want to dry off the water. I need a new pipette. So I'm going to get my tip, and I'm going to take my 10 to the negative 6, and I'm going to vortex. Take the cap between my pinky and ring finger, flame the opening, put the tip in, slowly release my thumb, flame it, cap it, take my melted agar deep, flame the opening, add it to the agar. Okay, flame it, recap it, get rid of my tip. I need to add my E. coli to this tube, so again, I'm going to vortex. I'm going to get my transfer pipette again. Every time I go into the tube, I've got to flame the opening. So flame the opening of the tube. Go in and draw up 0.5. Okay, and I can release it into the pipette. I don't have to hold it there. Flame it, cap, add it to my agar deep. This time it doesn't matter if I touch it because I'm on my last one. And I should have flamed it before I put the lid on it, but I'm going to flame it again here right before I pour it, so I didn't. I'm going to turn my 10 to the negative 6 plate over. I'm going to vortex this to mix it. And I'm going to flame the opening of this tube. And I'm going to let it cool for a minute, but close to the flame, because again, my flame is my sterile field. So I'm going to let it cool for just a minute, and then I'm ready to pour it. So I'm going to pour carefully onto that plate. Put the lid back on. Put the cap back on. Now I have poured all 4 of my plates. So all 4 of my plates have the same concentration of E. coli. The E. coli is just there as the host cell. And then I have plated my various serial dilutions of my phage stock, because my phage stock is what I'm trying to determine the titer. So I have my 10 to the negative 4, my 10 to the negative 5, 10 to the negative 6, 10 to the negative 7. So I need to leave these plates to solidify. It takes about 20 minutes or so. I want to make sure that the agar is completely solidified before I pick up those plates. So once I let those sit for 20 minutes, what I'm going to do after they've solidified is I'm going to take all 4 plates and put them in a stack. I'm going to rubber band them together, and then I'm going to flip them agar side up and put them in a 37-degree incubator. And so I'm going to incubate those and then use those plates to determine my titer. And so this is how we do the setup for the plaque assay.
So this is day 2 of the plaque assay, and in this day we're going to do the readout. So when we do our plaque assay, again, the purpose is to determine the titer, and our units for titer are going to be PFUs per milliliter. PFU stands for plaque forming unit. We'll talk about what that means in a minute. So our purpose is to determine the titer of an original phage stock solution. And so again, a phage is a type of virus that infects a bacteria. And the way that this works is that we have our bacterial host cell. So here is our bacterial host cell. And in this particular experiment, we're using a specific strain of E. coli called strain B. And this particular strain of E. coli is very sensitive to phage infection, meaning it's more likely to be able to be infected by this phage. So we have our E. coli that is strain B, and we have our little phage. And our phage look like these little, spaceships. And so the way that these phage replicate is that they land on the host cell. So they land using these tail fibers on the host cell, which in this case, again, is the E. coli. And when they land, they will inject their nucleic acids, their DNA, into the host cell. So they inject it into the E. coli. Now again, viruses are obligate intracellular parasites. They can't replicate outside of a host cell. Instead, what they do is they take that host cell and they make that host cell into a virus-producing factory. So when they inject their DNA into this host cell, it's going to hijack the host cell machinery, and the host cell is going to begin to transcribe this DNA, so make mRNA. It's going to make viral proteins. And so you're going to start to get all the pieces of the virus being made inside the E. coli. Once all the pieces are made, the viruses will be assembled. They're going to be put back into their functional, what we call virions. When we get enough virus inside this E. coli cell, and there's no more room, the last part of the virus life cycle is going to be to lyse that bacteria. It's going to cause the bacteria to rupture. When the bacteria ruptures, the phage are going to come out, and all of these new phage that are made are now ready to go infect another host cell. And so what you're going to see is when this happens in the bacteria, when the virus infects this bacteria, and it then causes this bacteria to lyse, we end up on our plates with what we call a clear zone. A clear zone is an area of cell death that's known as a plaque. And that's because the virus killed the bacteria. And so we end up with these clear zones or these plaques on our plate. And so let's take a look at that in a little more detail to understand how do we get these clear zones. So if we look at our plates, we end up with 4 plates, and our 4 plates are going to have different amounts of plaque. So if I look at this, and here is my experiment. So here is my original bacteria that was infected with a phage. Remember that we put so much bacteria in this, and when we put so much bacteria in this, we end up with a lawn. So we end up with all of these bacteria just next to each other side by side. So for every place a phage lands, that phage is going to infect that bacterial cell. That bacterial cell is going to undergo its life cycle of the phage. And remember that the last part of the life cycle is that once this host cell has made its virus particles, it's going to rupture and it's going to release those phage. Those phage can now go and infect adjacent cells. Now these cells are going to lyse and they're going to infect neighboring cells. So notice these cells all lyse and now they're ready to infect neighboring cells. And so what we end up with is that we end up with these plaques or these clear zones. And that's because each plaque, so if you look at this plate, this haziness is the bacteria. Each of these little clear zones represents one initial virus that's on this plate. And that one initial virus that's on that plate infected one bacterial cell, and it replicated and it caused cell death to other bacterial cells around it until we get these plaques, these clear zones. So if you think about it, when we do our 4 plates, we have a 10 to the negative 4, 10 to the negative 5, 10 to the negative 6, 10 to the negative 7. We have 4 dilutions of our phage that we are plating. Which one are we going to expect to see the most plaques? Which one do you think you're going to see the most clearing? Which dilution factor? If you said 10 to the negative 4, you would see the most plaques. You are correct, because your 10 to the negative 4 is the most concentrated of your virus. It's the highest concentration. It's the lowest dilution. So this would be the equivalent of our 10 to the negative 4. This would be our 10 to the negative 5, 10 to the negative 6, and 10 to the negative 7. And so we would look at our 4 plates. And just like our standard plate count, when we're determining our titer, only one of these 4 plates is going to be used. And so let's look at how do we determine our titer. So let's talk about how do we calculate our titer. So first we need to talk about PFUs. PFU stands for plaque forming units. It's the number of plaques that are found on the plate, meaning the number of clear zones. And remember that for each plaque or each clear zone that we have on the plate, that represents one initial virus that was plated on that plate. So what we need to do when we want to calculate our titer is we need to count the number of plaques on a plate that has between 30 and 300 plaques. You may recall that during the standard plate count, this was very similar. When we did our standard plate count, we were doing colony forming units. and we were counting the number of colonies on a plate that had between 30 and 300 colonies. Similar idea here, except the only difference being that we're not counting colonies, we're counting plaques, because the colonies are bacteria and the plaques are for the virus. And so remember that on our plate, we have a lot of bacteria, and what we're looking for is that we're looking for those clear zones or those plaques, and that represents one initial virus. So we need to count our plate that has between 30 and 300 plaques. So if we have less than 30 plaques, we have too few to count. If we have more than 300 plaques, we have too many to count. So it needs to be the one plate that has between 30 and 300 plaques. So if we are going to calculate our titer, the way we calculate our titer is that titer is equal to PFUs, so plaque forming units, divided by your final dilution factor. That's the final dilution factor that's written on your plate. So for example, let's say that we had 115 plaque forming units on our plate that was labeled 10 to the negative 6. So we have 115 divided by 10 to the negative 6. That is equal to 115 times 10 to the positive 6. So when we divide by a negative exponent, it's now multiplying by the positive exponent. So it's 115 times 10 to the 6. That is not in proper scientific notation though. We want this first number to be a number between 1 and 10. So my decimal point is here. I need to move it 2 places to make this 1.15, which means my exponent needs to move 2 places. And so what we end up with is our calculation is 1.15 times 10 to the 8th. Notice it moved 2 places because I moved my decimal point 2 places. So 1.15 times 10 to the 8th. And then my units are going to be plaque forming units per milliliter. So this is how we calculate our titer. Again, it's very much the same idea for how we calculate standard plate count. But in this case, instead of counting colonies, we're counting plaques. But the math is essentially the same. So what I want you to do is to look at these plates. This is our 10 to the negative 4 plate. These are the plaques. Again, remember that my 10 to the negative 4 is going to have the most plaques on it because it is the most concentrated or the least dilute. And so you'll notice that this one has way too many plaques to count. That one can't be counted. 10 to the negative 5, this plate has greater than 300. 10 to the negative 6, if we count each of these little plaques, whether they're small or large, every plaque counts. If we count those plaques, we get 84 plaques on this plate. 10 to the negative 7 has 5 plaques on this plate. And so you can see that only one of those plates has the correct number of plaques in order to determine the titer. And so what I want you to do is to calculate your titer based on this image, and we can discuss this in the Zoom to check your answer. So use this image, calculate your titer, and then we can discuss it in Zoom and make sure that you got the right answer. If given a question like this on your exam, the number of plaques on the plate will be labeled. You will not be expected to sit there and count the number of plaques. They will be labeled for you, just like you're seeing in this image here. You just need to know which plate to use and then how to calculate that titer. And so this now concludes our plaque assay.